Hello and a warm welcome to all biotechnicals. In my earlier video, I had discussed about the most important topics to study from GATE Biotechnology subject. Today, I'll be doing the same for GATE Life Science paper. As you would all know, GATE Life Science has a compulsory chemistry paper and 5 other subjects: Biochemistry, Microbiology, Botany, Zoology and Food Technology, from which you have to select any 2 for your exam. But today I will discuss all the important topics from all of these papers so that you get a consolidated list in just one video. Don't forget, you have to prepare for only 2 out of these 5 subjects in your main exam. Let's begin. Starting with chemistry. This subject has a total of 9 sections. I'll be telling you for each one of them. From section 1, you must focus on Planck's quantum theory, Bohr's model, Hund's and Pauli's exclusion principle, periodic properties, ionization energy, electron affinity, electronegativity, and atomic size. From section 2, different types of bondings are important like ionic and covalent, molecular orbital and valence bond theory approaches, the Vesper model, terms like hybridization, resonance, dipole moment, different bond structural parameters, and other stabilizing interactions like hydrogen bonding, Van der Waals interaction, ionic solids, ionic radii, and lattice energy is also to be studied. From section 3, which is sp and d block elements, study the general characteristics of 3D elements and the coordination complexes, from which study the valence bond and crystal field theory, geometric magnetic properties, and isomerism. From section 4, which is chemical equilibria, osmotic pressure, elevation of boiling point, depression of freezing point, hydrolysis of salt, buffers and their principles, pH and HH equation, which is Henderson and Hasselbalch, equilibrium constants, learn the terminologies Kc, Kp and Kx, learn the different formulas involved. From electrochemistry, important topics are conductance, EMF, standard electrode potential, Nernst equation and thermodynamic aspects along with their applications. Section 6 deals with reaction kinetics, rate constant, order of reaction and kinetics, molecularity, Arrhenius equation, activation energy, catalysis and enzymatic reactions, and reversible and irreversible inhibition of enzymes are important. Let me remind you, in this entire list you will find many topics which are common with the other subjects. However, you may not be preparing that subject, but yes, there are few common topics from other subjects too. Section 7, thermodynamics. First law, internal energy, enthalpy. Second law, entropy and free energy. Heat of reaction and Hess's law. Gibbs-Helmholtz equation, free energy change and spontaneity. Equilibrium constant are important topics. Section 8, structure, reactivity, correlations and organic reaction mechanisms. Acid and bases, their ionization, concept of aromaticity, stereochemistry, optical and geometric isomerism, tautomerism. From this concept, optical isomerism, chirality, enantiomer, diastereomers, and RSL's convention to Fischer projections are important. Next is elementary treatment of SN1, SN2, E1, E2 and radical reactions, Grignard's reagent, aromatic electrophilic substitutions, and identification of common functional groups by chemical tests. The last section, chemistry of biomolecules, is entirely important because anyways this entire section is the biochemistry part, which anyways you will have to study if you are opting for this biochemistry paper. The topics that you should cover here are amino acid, proteins, nucleic acid and nucleotides, peptide sequencing by chemical and enzymatic proteolytic methods, DNA sequencing by chemical and enzymatic methods, carbohydrates up to hexose only, and lipids, triglycerides only, principles of biomolecule purification. Here the common topics are techniques with other subjects like ion exchange and gel filtration chromatography, and also regarding spectroscopy, learn the Beer-Lambert's law and identification of these biomolecules. Coming to biochemistry. The first section deals with amino acids, where you have to study the properties and structures, important points and critical features involved, like amino acid with 2 chiral center or what is pH and pKa, the isoelectric concept application. Expect numericals from this area. Don't forget the classification. From protein structure and function, characteristics of secondary and tertiary structure, collagen, hemoglobin and myoglobin is important. Carbohydrates, concept of epimer, anomer and different examples of polysaccharides, including their examples and also the bonding which is present between them. From nucleic acids, purines, pyrimidines, differences between different forms of DNA, that is A, B and Z, and also you have to study characteristics of all this possible, especially for bDNA. Proteolytic cleavage of different enzymes. For example, chymotrypsin, ribonuclease A, like what are the important amino acids which are present in their active site. Also protein folding, where you have to study Levinthal's paradox and Anfinson's experiment. Section 2 covers enzymology. Enzyme kinetics and inhibition is what is most important. Obviously, you can expect numerical type questions from here, and inhibition is also a must do. You can expect both numerical and graph based questions from here. And while covering the kinetics, cover MM equation, LB plot, terms Km, Vmax, formulas and graphs. From metabolic processes, study the steps and regulations which are involved, especially the ATP calculation, the stoichiometry, and also the thermodynamics of the reactions, which is favorable, which is exergonic, etc. You can expect numericals from this area. Other important topics are nitrogen fixation and vitamins and coenzymes. From high energy compounds, study the examples, the standard free energies of different compounds, for example, phosphate hydrolysis, ATP hydrolysis, and also the standard reduction potentials of biologically important half reactions. You can easily find a consolidated list in Leninger. Don't forget to work on the numerical problems like calculating molar stock and working solutions. Section 3 is all about techniques. The principle and the application is what you need to study. Chromatography, all different types, ultracentrifugation, PCR, ELISA microscopy. From UV spectroscopy, study the Beer-Lambert's law and expect graph-based questions on absorbance. From DNA-protein interaction techniques, EMSA and DNA footprinting must be studied. Mass spectrometry and other protein-related techniques. From here, SDS page, isoelectric focusing and 2D gel electrophoresis. And protein structure determination techniques. For example, to study the 2D structure, how you will use circular dichroism, or for studying the 3D structure, you will use X-ray or cryo-electron microscopy or NMR. You need to know all of these principles and working techniques. From section 4, cell organelles function and cell membrane composition, charges of phospholipid, different transport types and the proteins involved, and cell signaling, entire molecule components which are involved like ligands, receptors, secondary messengers, etc. And cell death, programmed cell death or apoptosis is also important. Next is from section 5, inhibitors of translation, transcription, translation, as this is the entire molecular biology part. From here, antibiotics and mode of action is extremely important. Wobble hypothesis, enzymes and proteins involved in all these processes like DNA polymerase 1, 3 or clinofragment, rho factor. You can expect direct match the following questions, and also simple to the point questions are expected. Operon concept, lac-operon and tryptophan, DNA damage repair, and from recombinant DNA technology, the different types of vectors and restriction digestion is to be studied. The last section covers immunology, so cells of immune system and their function. Again, here you can expect match the following type of questions, different classes of antibody and their functions, properties of MHC and their antigen processing pathways, immunotechniques, ELISA and RIA. You can follow QB for details. And hybridoma technology, a sure short topic for GATE life science exam. So definitely a must do topic. Let's move into our next subject, that is microbiology. Here we have a total of 11 sections. First of all, let me tell you that this first section, historical perspectives, then section 4 regarding prokaryotic cells, Section 6, which is control of microorganisms, and Section 7, that is metabolism, questions are not seen much from the microbiology subject point of view. However, metabolism overall, you may see some questions, and sterilization, it may be important from the, if you're looking from the food technology point of view. But this is only regarding microbiology 1, 4, 6 and 7. Not many questions come from here. Let's begin with the section 2, which is methods in microbiology. Here microscopy is important and the different staining techniques like acid fast, gram staining, study the process, principle and mechanisms involved. From section 3, the Baltimore classification of viruses, structure, genome, etc. Study the corona one also. Archaebacteria and eubacteria, how they are different from each other. The classification and nomenclature of different microbes and also mycoplasma. When I say nomenclature based on different conditions like extreme temperature bearing or extreme salt bearing, and classification regarding the photoautotrophs or chemo, photolitho, like who will use carbon source or what they will use as energy source. On these basis, this particular unit is very important. Section 4, bacterial structure, which is regarding the cell wall, cell membrane, then flagella, cilia, pili, ribosomes, mesosomes, these all need to be studied. And also the differences between the prokaryotes and the eukaryotic cells. From section 5, that is microbial growth, growth curve is important, growth kinetics, that formula which involves nt is equal to n0 into 2 to the power n. You can expect numerical questions from here. Then doubling time and also the graph based questions can be expected. Something regarding little bit about culture media, the differential media, specialized, general media and also common media examples, right? Then from section 6, yes, different sterilization techniques, you can study it in general, not in much detail. Then section 7, fermentation and also bacteriochlorophyll, what are the different features you can study. Then again, this is an important topic, that causative pathogens and the vectors from section 8, which is regarding the disease and host pathogen interaction. So as you can see, the third unit and the microbial growth and this 8, 9 unit, these are very important from microbiology point of view. So from here also you can expect the match the following questions, main diseases, pathogens and also which vectors are carrying them. Under toxins, diphtheria, pertussis, botulinum, tetanus, these are to be studied. Some more also. Then from chemotherapy, antibiotics, again match the following, the different antibiotics and their mode of function. From microbial genetics, yes, the entire bacterial genetic system, which involves transformation, conjugation, transduction, recombination, then plasmid, transposons, all these concepts must be clear. And from microbial ecology, study the nitrogen fixation. As you can see, I mentioned to you many topics are common. Like nitrogen fixation, you had studied in biochemistry also under the second unit. So like that, you will find many topics which are actually common. Now let's move to the other subjects. First, we will be talking about botany. This subject has total of 9 sections, out of which section 4, 5 and 6 are the most important. Section 2, 3, 8 and 9, they are medium important, whereas not many questions come from economic botany and plant systematics. So maybe if you can skip if you don't have time. From the first section, the APG system of plant classification, the different characteristics like angiosperms, gymnosperms, bryophyte, monocot, dicot, this is what is asked if at all. And phylogenetics and cladistics also has questions. From plant anatomy, anatomy of root, stem and other plant parts, the special features and also section cutting. Plant families such as floral structures and conditions and their unique characters. Floral organs and floral formula. The vascular bundles like xylem and phloem structure and also anomalous secondary growth. From the third section, plant development and cell and tissue morphogenesis. Life cycle of angiosperm is important, like from the start to seed. Also the development of male and female gametophyte, embryogenesis, double fertilization, photoperiodism and vernalization, the ABC model of floral organ patterning, You have to study the different effect of mutations and also diagram based questions. Then from photomorphogenesis, phytochrome, the red or far red light, what is the impact on flowering, germination. You can expect some graph based questions also, concept and analysis. Then cryptochrome as well. Then from plant hormones, you have to study all the components involved like signaling, receptors, function, and also the biosynthetic pathways. From section 4, apoplastic, that is plant physiology and biochemistry, apoplastic and symplastic transport mechanisms, again nitrogen metabolism, which will also include fixation, the process and the enzymes involved, then photosynthesis, C3, C4, CAM cycle, photorespiration, electron transport chain. These are common topics with other subjects and important as well. Structure and function of biomolecules, amino acids, enzymes, NATs, about them, and major plant secondary metabolites like terpenes, etc. From section 5, which is genetics and genomics, you can expect cell cycle, principle of Mendelian inheritance like linkage, then extra chromosomal inheritance from their chromosomal and mitochondrial, then chromosomal aberrations, numerical like euploidy, aneuploidy, and even structural like deletion, duplication, inversion, translocation, and transposons. Then we have dihybrid cross. From here 2-point, 3-point based cross is a must. Definitely question will be there. And percent probability numerical type questions can be expected. From section 6, which is plant breeding, genetic modification, and genome editing, you can expect agrobacterium mediated gene transfer. Again, this is a must topic like where genes and functions. Then male sterility, genetic maps, molecular markers, haploid and double haploids, also the different selection markers like kanamycin, hygromycin, like Bt cotton, which gene will be modified, or let's say herbicide tolerant crop, soybean, which pathways they will inhibit, then beta carotene rich rice, which enzyme is affected, and also the different transgenic crops. Many questions can be expected from this segment as well. Then section 7, as I said, it's not that important. From here you can study about the different medicinal plants like alkaloid, flavonoid. This part is important, like which chemical from which plant it will be secreted, and also from which plant part. Okay, so match the following. You can expect, you can just study some plant parts, what is given in book, like some 30 medicinal plant and also some additional plants from the previous year question paper. Don't forget to study the botanical name as well. Then cereals, beverages, oils, rubber, plant yielding fibers, spices, again from which plants they are, plant part they are extracted, and what is the botanical name. Then from plant pathology, the plant immunity, that is systemic acquired resistance, ISR, PR genes, plant diseases, different fungi and bacterial both. Match the following, multiple select question based on their classifications. What you can do is you can solve them through option elimination also, right? Molecular detection of pathogens, the different causative agents, and also the salicylic acid and jasmonic acid pathways, host parasitic plant interactions. From the last unit, mutualism, niche, habitat, ecological succession, pollution, global climate change, and speciation extinction has some amount of questions. Let's move to our next subject, which is zoology. Coming to the first section, which is regarding animal diversity, you can study the classification of animals. Basis of classification like body symmetry, body cavity, circulatory system, etc. Examples of animals in different phylum, the representative animals, and also what are their special characteristics. Phylogenetic relationships based on classical and molecular phylogenetic tools. Then evolution, theories of evolution, natural selection, examples graphs, adaptation, example of homologous or analogous organ. Then what are the different types of speciation? And most important is Hardy-Weinberg principle. Definitely question, numerical question will be there. Section 3 is genetics, which is common with other subjects also. Again, 2-point, 3-point cross is very important. Dihybrid cross numericals. So first you can study the basics of Mendelian genetics, and as I said, dihybrid cross numericals. What is incomplete dominance, codominance, multiple alleles, Then gene interaction ratios, these all will come for match the following. Then extensions and deviations, what are the examples of sex-linked trait, sex-limited traits and sex-influenced traits. Then genomic imprinting, from linkage and recombination 3-point cross, gene order determination mapping has to be studied. Then examples of genetic disorders, expect match the following, like heritability and therapeutic factors. Also the sex determination in Drosophila. From section 4, biochemistry and molecular biology, this is again, some topics are common with chemistry subject anyways, which is compulsory. Nucleic acid, proteins, carbohydrates and lipids. Enzymology, kinetics and inhibition. Glycolysis, TCA cycle, the regulation and ATP calculation. You don't have to go in as much detail if you are preparing for biochemistry. So this would be like how you are preparing for chemistry. And the central dogma of molecular biology, the important steps and the proteins which are involved. From cell biology, this is also common with biochemistry. Here, cell cycle, basic concept, cell cycle and cell division. Then what are the function of cellular organelles like cytoskeleton, motor proteins, what is the organization. Then chromosomes, chromatin structure, DNA content of the cell has to be studied. Microscopy is an important section here. The types, Abbe's equation and basic principle of different types of microscopy. Again under cell signaling, the different ligand, receptor and effector. Here you should focus on the signaling molecules which are important from development point of view, especially the WNT pathway, hedgehog signaling, sonic hedgehog signaling. Apart from that, GPCR and RTK is also to be studied. Then gene expression in eukaryotes, you have transposable elements, for example line, sign, ACDS elements, expect match the following here. Prokaryotic gene regulation regarding the lac operon and tryptophan operon. Then from section 7, animal anatomy and physiology, endocrinology is most important. Here hormones, biosynthesis, mode of action, target tissue and exocrine glands are to be studied. From respiratory system, cardiac output and respiratory volumes, you can expect numerical. Then reproductive system, digestive system and excretory you can also study to some extent. Parasitology and immunology, section 8. Here disorders and diseases, again you can expect match the following. Example, what is the host, parasite vectors like sleeping sickness, trypanosoma brucei. What is the host vector, primary tissue, or what is the organ of residence, everything. And household insect vector and the associated disease like sandfly, kissing bug, etc. Then also yes, the different causative agents of disease disorder. Then you have the antibody functions of the immune cell. There you have antibody functions and hypersensitivity, autoimmunity diseases and their detection tests like Vidal, which is for typhoid. These also questions can be seen. Yes, being zoology, development biology is a part of it. So first of all, Drosophila is important. Drosophila developmental genetics. The different maternal and zygotic genes and also the axis development formation and what is the effect of the mutants. What will be the effect on the phenotype. Then we have fertilization, how the zygote forms, how the morula forms, how the blastula forms and what are their special characteristics and process involved. In gastrulation, the different type of cell movements like invagination, involution, any important terminology which is related to these processes has to be studied properly. Also the order and sequence of these events. Then stem cells and developmental signaling, which I said is common with the signaling pathways. Section 10 is about ecology. Here you can expect numericals from Lindman's law of trophic efficiency. Under conservation biology, you can expect questions on examples of endemic and endangered species like factual information and also in or ex situ conservation. From biogeography, you can study hotspots and different type of species like keystone, generalist, these are important. Animal behavior, behavior and types, that is hibernation, estivation, round dance, and also the different ecological concepts like kin, spite, niche, habitat, altruism. You can expect match the following questions from here. The last subject is food technology. I will not be discussing much in detail from here because this has only 4 sections, but then these are complete full paragraphs. So overall, these are the topics which are important. Remember, momentum transfer, heat transfer, mass transfer, mechanical operations, thermal operations, mass transfer operations. Okay. Apart from that, kinetics, growth kinetics, toxins, fermented food beverages, milk and milk product processing, oil processing, and the principles which are involved are very important. I hope that this has cleared your doubt regarding what are the important topics of GATE Excel. If you want any other aspect of GATE Life Science to be discussed, then please do drop a comment. Thank you.